for the rest of the time here, the experimental design thing was just kind of a little appetizer for you. Now we're going into the sort of more bioinformatics part of the day, which we'll do for the rest of the morning. And I will start by giving a few slides going over what multiomics data is and a brief overview of the different methods that you can use to analyze it. Of course, we're only going to kind of look at one in our workshop, but there's other things out there just to make you aware. And then we'll do the hands-on stuff. So let me do these intro slides here. Yeah. So yeah, we're kind of talking to you as these little workshops that I do are not going to teach you everything you need to know, right? Like this is something that you can take multiple classes over multiple semesters to learn about. This is just sort of to give you an exposure to a few things and a big picture view that then gives you like the jumping off point to like, if you really want to sit there and learn how to do this stuff, you now have just enough knowledge to know where to get more knowledge, basically. So that's like, yeah, I'm not gonna promise you're gonna become an expert in like 2 hours, basically. So sorry about that. But before we actually start, these links will be in the slides that you can look at later. There are some great resources out there for learning about multiomics, and so I'll just go over them. There's the American Statistical Association has a short course on statistics and genomics and genetics, which includes some of the multiomics stuff and a series of lectures. They have it like in text form and video form, so that's a good one. Mixomics, that's basically one of the main packages in R that we are going to be using over the next couple of days to analyze our omics data. And there's sort of a whole group of people that have developed this, and they have a bunch of like webinar video things about it, and periodically they offer these sort of like asynchronous online courses that you could take. So if you're you should look at their schedule there. They also have like basically a textbook they wrote that you could go through as well. And when I did a previous set of omics workshops, I based my materials off of this class that was offered in Sweden for they unfortunately if you're not from the European Union, you can't actually take these courses directly, but you can look at all their materials. And I mean, but here's the thing about the omics stuff: the methods are changing so fast that it's like, well, is this class from September 2021 still the state of the art? You know, some of the stuff they did back then, people are still doing, but there's probably a bunch of new. I can't say that I know the full state of the art of, you know, all the new stuff that's coming out. So, you know, like the world's moving fast these days. So who knows if this stuff still, but yeah, stuff from a few years ago still works, but there might be something newer and better out there. Yeah, so you'll have those links. Take a look at those if you want to learn more. So let's go over conceptually, big picture, what we're going to do. Again, please ask questions if anything is unclear to you. Yeah. So what are multiomics data? What do they look like? Well, it's basically, omics is a broad term for any kind of like multivariable data set that's collected from a biological organism. And so there's lots of different ones. You could have genomic data, you know, information about a bunch of different genetic loci. You have sort of methylome, looking at patterns of methylation at different loci in the genome. Metabolome, cells or organisms produce many compounds, metabolites. You can measure the concentration of each one. So there's many variables there. Transcriptome, you know, how many different kinds of RNA are floating around. Obviously, there's proteome as well to see what's actually being expressed, and then microbiome. many species of or taxa of microbes in an organism or in an environment, and we can characterize the abundance of each one. So yeah, it's anything you have a lot of different variables or features from. And yeah, as it could be at the single cell level, it could be at the organism level, it could even be at a broader level, environmental omics data as well. And whenever we have omics data, we also have other metadata or covariates about these organisms or cell or whatever it is, whatever experimental or observational units we collected the data from. If you all are doing designed experiments in the lab, treatment is obviously going to be one of these variables. You got genomic or gene expression data from your high fat diet and your low fat diet and everything like that. Obviously, individuals also have covariates like your size, sex, all of that, and then you have outcomes as well. We want to relate the omics data not only to the treatment but also to the outcome. Disease status or survival or things like that. And obviously, if you have single cell data, you're also going to have their covariates on the individual cell, like cell types or other things you might be looking at. So what can we use omics data to, what science questions can we use? Well, we can look at relationships within the omics. What is the relationship between, a microbiome and metabolome? Are different microbes stimulating the production of different metabolites in a cell or in an organism? And then all of that sort of put together, how do those omics data predict the outcomes that we care about? Like, is there a microbiome signature in obese versus not obese individuals, if we're concerned with obesity and things like that? And okay, so now let's talk about some of the different techniques that we might use to analyze omics data and how we can group them. This is kind of a broader idea from, machine learning, not just for omics. There's kind of two main types of methods to look at multivariate data, which is supervised versus unsupervised. Supervised means we have an outcome variable that we already know, like let's say it's disease status, like people with heart disease or that had a heart attack versus people that did not. Can we then look at their gene expression or other omics data they have to predict heart attack? So then we could be like, okay, we can take omics data on people and then later predict if they're likely to have a heart attack or not based on just the omics data. That would be like a supervised one because we already know the groups that the different individuals belong to. And we're trying to use the omics to separate those out. Unsupervised is where we don't necessarily have an outcome. We're just seeing if there are clusters in the data that we can find based on the omics. And so then it's like, well, once we identify those groups, we can then sort of tell stories about what those different groups might be. But there's no predefined outcome that we're trying to see whether the omics align with. We're just trying to see if there's patterns in the data, if some samples are more similar than others, that we can draw kind of these clusters around. And within your supervised methods, there are two types of analyses that we can do: descriptive and predictive. Descriptive means we want to find weights, sort of like estimate parameters for the model that optimally separate the classes. And what defines optimal, that depends on, there are many different ways to define that. But basically, that would be like, descriptive would be in your designed experiments, you probably do more of a descriptive analysis where you're going to say, okay, we have, individuals that have a controlled diet versus a high fat diet. Can we use omics to separate those out. But predictive would be like in that heart attack example, like I said, if we collect new omics data and we know the omics for it, but we don't know what its outcome is, can we predict? So it's basically like then you can then like see how well you did by later finding out whether or not those people actually had a heart attack. You can then say, well, we were 95% accurate, or we were only 60% accurate in that. So that like predictive, you kind of get an answer of whether the model was good or bad. And there are various measures of performance. But really, you sort of need, in order to have a model that is descriptive, it also needs to have good prediction performance as well, because if a model doesn't make accurate predictions about reality, then we shouldn't use it to understand reality either, like we were talking about with experiments. Experiments need to have a correspondence to the real world. All models do to be useful. So what method do we use? But it depends on the size of the omics data set. Their omics data sets vary greatly in their sizes. So like metabolome, it's very expensive to measure the concentrations of metabolites, and there really aren't like that many different ones in a cell. So there might only be dozens to hundreds of metabolites in a metabolome data set, versus like your microbiome or transcriptome, you would often see thousands within an omics data set of different microbiome taxa, or of different types of RNA that are floating around. Genome or methylome gets even bigger, where obviously there are thousands to millions, depending on your sequencing of different loci you would have in the genomic data set. So that can be very big. Now, but big data is two-dimensional. So there's P, we were just talking about, that's the number of predictors, or the number of features, the number of different things in the omics data set. That can be very variable, like we just said, from dozens all the way up to millions. But N is your number of samples. So like your data is in an N by P matrix. And in clinical and biomedical research, right, N is not going to be super big, right? Like your studies on mice or humans are not going to have millions. They're going to have from tens to, at most hundreds of individuals. So sometimes you could have P could be way bigger than N, depending on the size of the omics data set.
Total samples are per group.
I mean, so yeah, that would be, I'm just saying this in rough orders of magnitude, usually N would be like the total number in the study, at least in this mission. But yeah, it's the number per group that really matters for determining.
Can you say some example for predictor for futures in the P? If that's something.
Yeah, so that would be like in a genomic data set, the P would be all the different loci. Each one would have would be a feature, or in a metabolome data set, each metabolite is a feature or variable. Yeah, so there's many of them.
Most of the time N is going to be smaller than P.
That's right. In these type of data sets, usually, yes. So yeah, basically, if you have small p and large N, that is where like very computationally expensive but gold standard methods like Bayesian work well. In a world with a little bit more predictors, you can use sort of classical statistics. When you start to get really, really wide data, then you need sort of more machine learning based methods to deal with the very, very high number of predictors relative to N. What do we use to do omics data in terms of software? Well, we're going to use R in this workshop, and a lot of bioinformatics packages in R are on a platform called Bioconductor. And so some of the packages we're going to work with come from there. A lot of the same stuff can be implemented in Python, like me and Matt were talking about. There's sort of sometimes particular models are only implemented in either R or Python, but nowadays it's relatively easy from within R to run Python code or vice versa. So if you have a special model you want to use, you don't have to switch around your whole software environment. And there's like also a lot of sort of graphical interfaces for omics analysis, like Metabo Analyst is like an online kind of point and click platform for doing omics analysis. Most focus on metabolome, but you can do other stuff with it. But it kind of runs R in the background, and you can get R code out of it. Yeah, so this is like one of these things that they say a data scientist spends more than half their time just cleaning and processing the data and organizing it, versus, you know, only a small percent of the time is spent doing the cool stuff of like actually making the statistical or machine learning models and learning things from them. Most of the time is spent just messing with the data. And I think that's probably no exaggeration, right?
An addendum to like the not addendum, I don't know if they're critic. It's like, you know, you're gonna have lots of features, and then what you're gonna see hopefully is a lot of times you're going to select for features that are meaningful. Like you're not going to put the whole data set into whatever, you know, statistical analysis that you want to do. You're going to go and do some sort of feature selection to clean the data first. And that's also represented in this graph, because
100%.
in the 60% of the time you're trying to find meaningful ways to, you know, bring your data to something manageable before you start running statistics.
Data from the raw data, the useful data.
Yeah, and then it's like, well, you know, I don't want low abundant features, you know, because that's a lot of noise. You know, it's not meaningful, particularly when you want to compare between groups. So you want to say, oh, I want to see only features that happen in, you know, they have to be present in at least 10% of my samples in order to be considered in downstream analysis. Things like that helps you shrink piece. Yeah.
And we will do both those things, both like all of that stuff in the sort of practical examples that we're going to do. So yeah, part of the data cleaning is getting rid of the features that have a lot of zeros or don't have a lot of, because if something doesn't have any variance, no variation in it, it doesn't have any information that can be useful. So, you know, if we want to know, does variation in X explain Y? If X doesn't have any variation, there's no way we can learn about effects on Y. So you may as well get rid of those. Obviously, omics data there's often missing values in some, you know, for whatever reason. And there are ways to kind of account for missing data, but if some individual, like if one mouse, the data weren't good or a lot is missing, we might just have to remove that mouse, or if some metabolites were only characterized in abundance for a couple of individuals, those aren't going to be very useful. So we remove those. But if there's sort of only a little missing data, we can kind of use some statistical trickery to fill in the missing data. We won't go into that today, but that's an important part of the process. So yeah, let's talk about just kind of how these multivariate techniques work in a broad sense before we actually do it. So basically, any of the methods we use is trying to find ways to represent a large matrix of data with high dimensions with a smaller number of variables that actually can convey most of the information in that big matrix in a smaller amount of numbers. That's like what all of them are doing basically. And the original method that was developed to do this, which actually has been around since 1904. So like people have been doing this stuff for a long time, is PCA, principal component analysis. And old as it is, it's still very effective and because it's old and relatively simple, it can be computed very fast, even on big data sets. So it's often a good first step to like visualize patterns. And basically what PCA is trying to do, it can help you understand the structure of the data and pick out biases or errors. What it does is it tries to approximate a large matrix, n by p, you know, n being the number of individuals or samples, p being the number of features in your omics data set by the product of a n by very small number matrix and a very small number by p. If you know from, your maybe even high school math that like if you take a n by 2 times a 2 by p matrix, the product of that is n by p dimensions. So we want to find those small number of dimensions that can capture most of the information in that really big matrix of the original data. That's more or less at its core what all these different methods are trying to do. So what are some of the more fancier methods for, and PCA is just looking at one omics data. It's not multi-omics integration yet. So yeah, there's a long list of multi-omics integration techniques. We won't cover them all, but this is just so you can be aware of what's out there. So yeah, there's a lot of machine learning kind of methods for pulling out information in high dimension data sets and reducing it to a small number. Yeah, I won't go into how they work, but they're sort of like I call it like PCA on steroids because it's like a PCA, but PCA only uses linear combinations, but these machine learning methods are very good at pulling out nonlinear patterns in the data. So it can like, if you give it a bunch of like coordinates of the world map, it will be able to like reproduce that structure and figure out where the continents are using a low dimensional representation. And they can be used across multiple omics to create a consensus that pull out dimensions that explain variation across more than one omics data set. Yeah, I mean, deep learning, obviously, in the era of AI is a big thing. We're not going to do any of that today or tomorrow, but yeah, the thing about these deep learning methods is they require very big data sets. They require a very large N to be, if you have a small N data set, simpler methods actually work just as well, if not better, than a really complicated neural network type of thing. So, maybe for single cell, it could be useful, but basically what some of these deep learning methods do is they do dimension reduction, something like PCA, trying to pull out small number of dimensions to capture most of the information, followed by clustering, trying to find patterns in the reduced dimension data set or groups of samples that are more similar to each other. And the good thing about these is they work well for nonlinear patterns. And what a lot of people do is they'll run their stuff through one of these deep learning algorithms, pull out the reduced dimension data from it, and then put it through like a machine learning visualization method to actually look at the pattern. So you kind of do both steps. Network analysis is also a big one. I know almost nothing about it, so I'm not going to really talk about it, but you can use, there's sort of a whole branch of math called network analysis that's used to find relations. I mean, some of the causal inference falls under this umbrella. Basically, you sort of like find for each sample, you find its neighbors that it's most similar to, and then for that next one, find the one that's most similar to until you have kind of a network structure. And then what people do is for each omics data set, you'll make a network, and then there are techniques you can use to combine those multiple networks into one network that represents the relationships across all the omics. Very cool stuff, but I know almost nothing about it, so I won't. But if you're interested in learning, I'm interested too. So let me know. So there's both. This is kind of, we were trying to decide whether we should go over what we're going to do or whether we should look at multi-omics factor analysis. We decided not to go over MOFA in this workshop just because there's only so much you can learn in a day and a half. But yeah, it's just like what we're going to do. It's more for exploring data, and again, it's just like just like PCA or anything, it's trying to find a reduced dimension representation of high dimensional data, but now we're using multiple omics. So like, it's taking these very large data sets from each omic, reducing it into a small number of axes, and then finding basically weighting factors for each sample across all the omics data. And yeah, but ultimately it's pretty similar to what we're going to be looking at. But the nice thing about this is that it built in, it can actually, because it's sort of a generative model, it can predict missing data in the process of fitting the model. So that's kind of nice. And then once you have your sort of different factors, low dimensional, like you might only have 2 or 3 factors, you can come up with biological interpretations for what each of them is doing. But that's also sort of what we're going to be doing with the Diablo approach that we're going to be talking about in the next day and a half. So yeah, this one, okay, so this is important to pay attention to because partial least squares discriminant analysis is the basis of the technique that we're going to actually be going through in the hands-on example. So you can see just like PCA, it's trying to take a large N by P matrix, reduce it to a smaller dimension representation while still keeping most of the information. But there's one key difference. It has this Y column as well. So not only are we trying to look at variation in the omics, we also have like an outcome variable, a Y, or it could be the experimental treatment group that we use as our Y variable. So it's not only looking at so it's a supervised method. Like PCA is unsupervised. It finds patterns in the data, but it doesn't have any idea ahead of time what groups each sample should belong to. The PLSDA does. You actually are giving it groups and trying to find patterns that align with the groups we already know about. Just like if we have an experiment, we know ahead of time what group everything belongs to because we assign those groups. So basically you, just like any other method that's like an ANOVA style thing, we're trying to find variables or weights from the model that maximize the variability between the groups and minimize the variability within groups so that we can identify patterns that in the data that are associated with difference between groups. Like if a particular gene is very, very highly expressed in the treatment group, but not expressed in the control group, that gene is going to be identified by the PLS method as being important for telling apart the groups. And so it's going to get a high weight in the model. But if a gene has similar moderate expression across all the groups, it's going to sort of drop out of the model because it's not useful for telling them apart. Would you say that's accurate?
Yeah, and so like too, like it turns out when you do the you just do the analysis on that omics, what you see as differentially expressed or differentially abundant will pop up in the multiomics analysis.
That's
There's
right.
going to be a consensus. Like the statistics are different, but like the features that will help you select or discriminate the groups will be very similar. So
That's right.
That
Yes.
often,
This here is just a single omics, but the multi-omics is kind of just a bunch of these stacked on top of each other and combined.
and it's like,
Yes.
what pattern between omic layers helps me, delineate the groups.
Exactly.
It's like a cross omics pattern versus one omics layer. So
Yes, so then the PLSDA is extended to sparse PLSDA, which is basically just what we just said, except that a bunch of the vectors in the small number by P matrix are forced to become 0. So we're trying to use even less information to represent the full data set. Yeah, so it's basically you basically sort of do a variable selection, which makes some of the coefficients go to 0. So we're saying like, okay, this, only a few of the genes in our genome, or only a few of the metabolites in our metabolome are have enough are being used to differentiate between the groups. And that like lets us better identify which ones are meaningful for differentiating the groups. And then, yeah, basically we'll talk about this more. I'll just skip over this part because we're going to go over it in the hands-on part. So yeah, so what we're going to be going over, we won't really get to it till tomorrow, but ultimately the multiomics integration method we're going to be talking about today is called Diablo, which is implemented in the Mixomics package, and it is a form of sparse partial least squares for multiomics data. Diablo stands for something, but yeah, don't really have to know that, but it does stand for something. And yeah, it's a sparse PLS across multiple omics, basically. But we'll talk more about it as we go. And yeah, another important thing to look at is sort of the differential expression analysis, but it can be used for other type omics as well. And we're going to talk about this in our single omics analysis that we're going to do today. We can kind of find it is not exploratory. It's actually like a hypothesis testing approach where we say, okay, these genes are significantly higher expressed or upregulated or whatever it is in the treatment group versus the control group or things like that. That's basically what we're doing. And it's more or less, it's not really a multivariate approach. It's just kind of like a separate statistical test done on every gene and then using a correction for multiple comparisons to, because if you have thousands or millions of genes and you test each one to see if it's significantly different, you would get a lot of significant ones because by chance some of them might be different. So we have to adjust our p-value threshold to account for the fact that we're testing many genes. That's basically so this is kind of more of a single variable analysis repeated over many, many genes with various like approaches applied to correct for that. So we're going to be doing some of that today. Yeah, and there's we're not really going to be using DC2, but that's one of the packages that implements it. So yeah, this is the last intro slide I have just to tell you what example data set we're going to be working on for the rest of the time. Basically, we got this. It's really hard to come up with good example data for these type of things, but this comes from a paper that's sort of like a case study to illustrate multiomics analysis, and it was accompanied by a data analysis protocol that they published. And we reached out to the author, Matthias, who actually provided us with even more example data for us to work with. So that was awesome, and we really appreciate his help. But basically, the data set comes from brown fat cells of mice, which are basically like part of I don't know that much about the physiology of it, but like when vertebrates get cold, they can break down these brown fat cells to release heat and warm up their bodies. So that's what, or at least mammals, and that's what these cells are doing. And so they sort of stimulated the process of breaking down the fat in these cells to generate energy, and then measured transcriptome data, metabolome data, and proteome data at 3 different time points on cells. Not the same cells, but some cells they measured it 0 hours after the fat broke down, some 4 hours, and some 24 hours. So we have basically our treatment group is actually time points in this case. We have 3 groups: 0, 4, and 24. So that's all the data that we're going to be looking at is from that. And so it's the thing about it is, we're looking at, obviously, if you like directly stimulate a cell to do something, you're going to see large differences in gene expression because you're like really messing with that cell. The type of effects you all are looking at is much, much more indirect, right? You're looking at like, if we change the diet of an individual, does it like change the gene expression of their offspring or something? These effects that we're going to see in this data are probably larger and stronger effects than what you would probably be doing in your studies. But it's good to have some sort of effects in example data so we can illustrate what it would look like if there are strong effects. So yeah, don't get too, don't set your expectations based off of what you see in this data set, because it's probably much stronger effects than you would see in your own omics work. But, yeah, that's just a caveat. So does anyone have any questions right now? Great. So we are going to now get everyone set up. The rest of the time, we're basically going to be doing for today, the rest of today and all of tomorrow, we're going to be doing live coding where I'm going to be like writing R code on the screen in front of you and explaining what it does, and you can follow along. So that's everybody hopefully has either got their account set up on the demo server to run the code or has installed stuff on their own laptop. Does anybody? Well, here, I'll just show you how you would, did anybody who wants to run it on the demo server, did everybody get the invitation link and create an account? Has everybody done that who wanted to do that? If anyone didn't, it's fine. You can do it now. It only takes a minute, but hopefully everyone did that.
Locally, right? Can we do locally too?
Yes.
Sorry,
So did you?
I have not done anything with the server, but I locally installed it my computer.
Great. So yeah, as long as you have R installed and did you install the packages that you need for okay, great. So let me see here. Let me switch the mouse. Okay, here we go. If you go to the workshop page, what you're going to need to do if you're running it locally is if you've already installed all the packages, you're going to want to open this fill in the blank code worksheet. That's basically just an R script file. If you download that from this link and then open it inside your R or R Studio, that's where you're going to be running the code off of. And it's a fill in the blank, right? So like if I just gave you all the code and said run this, you would just like highlight and get run and not understand what the code was doing. But if I gave you no code whatsoever, people often fall behind, like trying to type everything in exactly. So I'm striking a balance by giving you most of the code with a few blanks here and there that you have to fill in, basically. So that's how we're going to be doing it. So, okay, so let's walk through how you would. So if you create a deposit cloud account, if you go into it and sign into your account, I mean, I can see a bunch more things, but you'll be able to see at least this July 2026 Little Rock in your spaces on the left side of the screen. And once you're in, I see some people have already created projects, so that's great. So what you're going to need to do once you're in there is go over and click on New Project. So yeah, you have to be within this space to do this. So go over here, click New Project, and New RStudio project. So then it will take a second to get started. Okay, so here we are, and once you're in here, let's see if I can close this. There we go. Put it on full screen. Okay, is that like, in terms of the text, is that big enough for everyone to see it? Okay, so then once you're in here, if you're doing this on the Posit Cloud, you're going to see a folder called Worksheets in the Files panel on the right. You're going to click on Worksheets to open the folder, and then you're going to go open the Part 1 worksheet. And you see I have a folder Answer Keys. That's all of it with the blanks already filled in. But like I said, I highly recommend working along with me and filling in the blanks so that you your fingers learn it. It's better for your learning. Yeah, and hopefully, Matt, if people ever get stuck with anything, you can like go around and see whether they have a typo in their code and stuff like that.
Yes.
Okay, so open that up. Okay, so can everybody get to this point either on their, on the demo server or on their own computer? I'll give, a minute or two to make sure that everybody is good. Oh yeah, I should mention also, if you go back to the workshop, page, you can see I have part 1 lesson text here. If you open that, oh, that's not, yeah, that's the slides. If you open part 1 lesson text, all the stuff we're going to be doing, I have it explained out in text. So if you are a more verbal learner, you can read along with everything that we're going to be doing here in full text form. But I'm not going to go through that text in here. Just later, you can go through it on that version. Yes. If anybody on Teams is having yeah, can people on Teams do like a thumbs up reaction or a smiley face or something if you're all good and have everything the way I have it on my screen? Cool, I see some thumbs up there. Excellent. Wonderful. Yes. Need help? Are you doing it?
Yeah,
I did. Yes.
I did it and then I did it.
I see. Well, yeah.
It's an error.
Um,
Error base.
Well, if you already did it before, then I see you have only a comment and the so you didn't copy the whole line and you go back. Okay, so yeah, I don't know. Somehow you're like lost at the end of it. So but did you already do this or are you is it the first time you're doing it? Yeah, just copy one line at a time, maybe. Copy that line.
It's just like long term, I already have, because
Okay.
we usually have it last to like running even
Yeah,
the.
just go ahead because if it's already installed, the worst case scenario is give you call. Okay, let's see.
So you already done it.
Let's see for a second.
Yeah, like how does this work?
Same way as our,
What do you have already installed? Oh, it's not.
Let's see.
no need to go to see and get, like,
Get it on there.
so you can see all the library.
Yeah, it's already on there, so that should be good.
It'll just pairs here because all of the stuff, this is just to
Just make sure it's all good.
yeah, so that's because it's fill in blank. So yeah, so your package,
Okay, cool. Thanks, you should be good. It'll take a second.
so you can see what packages are loaded in on this page.
So this is all already like done.
Yeah, the environment is set up, yeah.
Okay, so we were just getting everybody up to speed here.
Wait, Quentin, I have a quick question.
Shoot.
How long do we have access to all these materials here?
So this demo server will only be available for a couple of weeks more after the after this. I should mention that this demo server is only for demo purposes. Don't upload your own data to this and do stuff with it, especially not USDA data, because that would be bad. It's only for example data. Like, plus it has very, very little processing power. Your own laptop can do this. I only have it so you can, it only has like one CPU per person and stuff like that. So yeah, you're only going to have this for a couple weeks. So if you do take any notes and stuff on these R scripts, you can always like download the file. Like if you edited this file and saved it, you could go and export it and then you could save it to your own computer. So like after this, yeah, but in terms of the slides and all all that on my website, I will keep that up forever. But you can't go back on this demo server anymore after a few weeks because I only, paid for 1 month of it, so.
Okay, thank
Yeah.
you.
Okay. So let's, in terms of the setup, I'm just going to, if you can, I don't know, if people are not familiar with our lines that begin with a pound sign or hash are comments. And so if you execute them, they will not do anything. Basically what we have here is, oh shoot, the bottom of the screen. Oh, there we go. Yeah, so if you're not familiar with R, this is the console. It is where we run commands and see output that comes out. This file that's open right here is a script. A script is basically just a text file that contains code that we can run, but ultimately it's only a text file. And when you want to run code in R, you can either type it into the console and hit enter and run it, or you can execute code that is in a script. Obviously, it's better to execute code from a script because you can save that text file and run the same code again later. So I may occasionally run a line of code or two in the console, but pretty much everything we're going to be running off this script. And the way you execute code from a script is if you click on a line and hit the run button or hit Control Enter on your keyboard, either one, it will run that line. So my cursor is on the line 13. I hit Control Enter, and it loads the code library or package called ggplot2 that we're going to use to make plots. You can also run multiple lines of code at a time if I go and highlight them. So yeah, follow along with me. Highlights everything up to where we start to load the data and hit Control Enter or click Run. And that will load all the code libraries that we're going to be using in this session, plus setting some plotting options and color schemes that we're going to use. Okay. Next, we're going to import the data that we're going to be working with, and we're going to be using transcriptome data. And the way that it was provided to us by the study author was already in an R object, which isn't, I would say, too common, but apparently he had collaborators that did some initial processing on the data and already saved it as an R object. So, that you may encounter that situation as well. And so it's a .rda object, which can be read into sort of a data frame, which is like a tabular form of data into R. Now, if you are running this on the cloud server, you can run line 33, which will load it from a folder called data, where I've saved the data. So you hit Control Enter, and it will load that transcriptome into our environment. You can see that if you load it, your data panel now says transcriptome, which is like a special type of data object for storing transcriptome data. Okay. But if you are doing this on your own computer, you're going to want to uncomment this line. In other words, delete the comment character in front of it and then run this, and it will load it directly from, I've posted the data on a website. If you could just run it from there.
Can you repeat that again? Yeah.
If you're doing it on your own RStudio, you go to line 34, delete. It had a comment in front of it. You're going to delete that comment and then run that line, and it will import the data directly from a website where I posted it. It's a pretty small object, so yeah. Everyone good there? Yes.
Have some errors on. It's saying that it's not able to implement one of these packages.
Yeah, putting
But it did.
Called
Line
Gblots.
34 did,
Yeah,
but these did not load.
I see. Well, then you'll want to go ahead. Did I not do that in the install packages thing? You'll want to just install the Gblots package then if it's not installed yet.
For locally, it's in the list. For, I guess, are you doing it locally?
On the software.
Yeah.
Well,
Did you run the installation for the, did you run the installation on the tutorial?
it's a.
Because
It's getting errors are probably not.
For now, you're not going to need the Gblots yet, but here, let me see.
take that out.
Let's see. No worries. So yeah, well, that worked. Got the transcriptome. So G plots didn't work. Yeah, so yeah, you just want to, we'll just go ahead and install it again. So for whatever reason, it didn't. So yeah, now if you run library G plots, it works. Sweet. And you got the data, so you're all good.
Yeah, sometimes you have to, if you restart R and clear your, sometimes it can be tricky. With packages, sometimes you just have to restart your R too.
Okay, thank you.
Okay, so yeah, where were we? Let's see. So we've all imported the data. Everyone good? You should see transcriptome appearing up here. Okay, so what we're going to do now is inspect the data and look at what we're dealing with. So one way you can, within RStudio at least, you can see how over here there's like a little triangle next to transcriptome. If you click on that, it will show you the structure of the data. It's like, wow, there's a lot in there. But there is one, within it, there's an object called counts, which is the actual table of, that's our actual N by P matrix of counts of abundance of RNA transcript from each of the samples for each of the genes. And you can see here I have 3 dots. Anywhere in the script you see 3 dots, 3 periods, that's a blank that you need to fill in. So we're gonna every time replace those dots with code and then run the code. So we're going to use the dim function, which stands for dimensions. And we're going to look at how large is our N by P matrix that we're dealing with. And you can see RStudio pops up some like suggested autocomplete, but you can hit escape to make it go away. Now I'll hit Control Enter on this line. So you can see we have 45,000 by 18. So we have, it's actually a P by N format. It has 45,000 rows, 1 for each gene, and 18 columns, 1 for each sample. We have 3 time points, and there's actually 6 samples per time point. 6 cells that were sampled at time point 0 hours, 6 at 4, and 6 at 24. And this is just some code to add the group column to our transcriptome data, because right now the data does not say which sample each group belongs to. I'm sorry, if you all are still, I don't want you to fall behind. So are you able to get everything going there?
Yeah, almost backed up. It's
Better to wait than to like have you fall behind. So.
Are you ready to vote? Very on. So. Should be good. Okay, your transcriptome. And I already added diff. Right now,
If you use this locally, then you have to install the packages, right? First.
That's right. Yes, so I set up the RStudio environment on the demo server to have the packages pre-installed and all the data there and stuff. But you wouldn't have that if you were doing it on your own. Yeah.
So I have a very stupid question. How do you know which packages to install if you are doing?
Well, yeah, I mean, you would. So that's basically R has like very little functionality base. Most of the actual functionality in R is in these packages, which like people have contributed to a library of packages. And so, yeah, so if you want to do a particular analysis, it's usually in a package, and you would have to kind of like, yeah, you'd have to know in advance what package you need to do that analysis.
Okay.
But, most people just copy and paste code from other places. So most code that you copy would say what package it's going to. So that's a starting point. Yeah, so, well, I'll just, before we're going to, basically the data that we got from our collaborators in this case does not say what, it just has like sample IDs. It doesn't say what group or what time point each sample comes from. So we're just gonna artificially create a column that says all the time points and tack it on to our data. So we're gonna, basically, we're gonna, in line 40, your lines might change if you added or removed lines, but it started out on line 40. We're gonna use the as.factor function to create a factor column, which is a data type that is used to identify groups in an experiment. So I'm gonna run that line. And then transcriptome has a object within it. This $sign means out of a larger nested data object, pull out a smaller object. So that's what the $sign represents in R. So we're going to, within the object of transcriptome called samples, we're going to create a new column in there called group. And this like left pointing arrow means assign the thing on the left or the right to the thing on the left. So we're going to take this groups vector we just created and create a new column called group in the transcriptome metadata that will say what group each thing belongs to. So I'll hit Control Enter on that line to run it. And now on line 42, all this line does is it's going to print the samples object and see what it looks like. So we'll see, we can just take a look at it. You can see that that the transcriptome samples has what group each sample belongs to. We just added that, and it has a library size. That's basically like the sequencing depth for each sample, and we're going to use that later to normalize because when you have transcriptome data, the number of transcripts you observe from each gene is a function of how many sequences total you got from that sample. So if we want to compare two different samples, we have to adjust for if they have different sequencing depths, different library sizes. So this is important metadata that we kind of have to be able to do that normalization. So you can see that the samples have some very different sequencing depth. Some are over 20 million and some are like 7 or 8 million. So they vary by a factor of 3 or so, which I would assume is fairly typical for this type of data, or is that like in the ballpark of what you usually see?
Most of the time it's not like for transcriptomics, you'll probably like, oh, I want a certain amount of data, and that's kind of your bottom line. But a lot of times you'll find some pretty big variation between the depth of the sequencing.
And so what would be like a minimum that you want?
For transcriptomics, I mean, the read counts for these are like 26 million, right?
Yep.
Is that what it says?
That's right.
Yeah, like I think generally speaking, you probably want over like 10 million.
Okay.
10 to 20 million.
So like a few of them are kind of borderline, but most of them are pretty good.
Yeah.
Yeah,
Also,
so there we
sample
go.
number. Samples.
So yeah, like, and sometimes this is where you'd be like, well, if one of them only had 1 million or something, you'd be like, well, that's like, that sample is useless and should be thrown out if it doesn't exceed the like minimum. So we're just continuing to look at the data and see and observe and explore it. So the next thing we're going to do is we're going to use the head. Head means give the first few rows of a data frame of a table of data and print them out, because we have 45,000 by 18. We don't want to see all 45,000 rows of output spit out here. We're just going to look at the first few. So if I hit Control Enter here, it will show the first few rows. So you can see that each row is named after ensemble ID of the gene, which is basically a numeric ID that doesn't really tell you anything, but each one has a unique one. And then these are the sample IDs. Remember, this doesn't tell us what group each one belongs to. That's included in the metadata. But each value in here is basically the read count for that particular gene in that particular sample. So, some are thousands and some are 0. Good so far? Okay, and now the good thing about working with mice is they're very well understood in terms of their genome. So we already have annotations that exist that will actually tell us what some of these genes are, like what protein they code for and stuff. So we can, there's actually an R package that contains that annotation database for mice, and we can pull out that data and associate each of these number IDs with an actual protein that the gene codes for, which may be useful for us when we're looking at which ones are differentially expressed later on. So that's what we're going to do now. So first, we're going to create a new a vector from the row names of this data. So these are like the numeric IDs. So we're going to say row names of transcriptome. Hit escape to get rid of the autocomplete and create a vector called gene ID from that. Hit Control Enter. And we want to, so basically, since we loaded this musculus package earlier, that loaded an object into our environment invisibly that contains the annotation database for the mouse genes. So we want to, this argument here on line 49, the keys equals gene ID. So we want to say, okay, find within this annotation database these numerical ID genes, and then key type equals ensemble, because that's the numbering system that we're using for these genes, and then it'll give us a column called symbol back, which is actually like the name of the gene symbol, which tells what protein it codes for. And so if we, this 47 through 52 is like a one line of code split up across multiple lines. So if we have our cursor anywhere within that and hit Control Enter, it'll run the whole thing. And it takes a second because it's pulling that information out of the data table. So it might take a few seconds, especially on the demo server is very slow. Does it return a one-to-many mapping between keys and columns? That just means that some of the numerical IDs have the same text ID, but that isn't a problem for us right now. So I'm going to run this line 53 to look at dimension of genes. And it has 46, we only had 45,000 in some genes, but there's 46,000 in this the annotations that we got, which means there's some duplicates. So the next thing we're going to do is get rid of the duplicates. And the way we're going to do that is by in R, the exclamation point means not, so it negates anything after it. So not duplicated is the function we're going to use. So any genes, any duplicate numerical IDs, we're going to get rid of from our annotation, and then we're going to replace that in our. That maybe isn't the best because there, if there's more than one gene with the same ensemble ID, we're just kind of like getting rid of the second one, which might be the one that has more read count, but we're just kind of going to ignore that little technicality for right now. Really, what you probably want to do is like go and find the one that has higher read count and keep it. But I think in this case, only a very few get removed, and it doesn't really make a big difference to the results. So just for learning purposes, we're kind of glossing over that. So I'm going to hit Control Enter to run that. And then we're going to add that annotation information to the object within transcriptome called genes, which contains the metadata for each gene. Hit Control Enter there. Okay. Yes, I see someone on Teams has their hand raised. Go ahead.
I think I heard you saying about how to deduplicate it in the real case. Is that to remain the higher reader counts? Is that correct?
Yeah, I think that would probably usually be a good idea. Yeah.
Okay, great. Thank you.
That. like to deduplicate, would you go in and get rid of the one with the lower counts every time?
It might be safer to just make the names unique
Yeah,
and then run the data and then maybe like assign a new name when you do the flush.
That's right.
Because if it's important, it's
That's
better
right.
to
That's
be,
true.
yeah, like you can just like make the names unique as opposed to.
yes. So yeah, you could just like assign a new numerical ID from 1 to however many genes there are, which would ensure there's no duplicate IDs, and then do the annotation after the fact. That would make it better.
Okay, thank you so much.
So now we're going to do some transformations on the data, which we have to do before we are actually trying to compare expression between our time points. So I mentioned before that these samples, we saw in the metadata that the samples have different total read counts in each sample. Some had. Do people need help with their with stuff?
Yeah,
I can-
it's back when they're having problems with the annotation DPI.
I see. The package wasn't installed correctly?
No, it was you type, I think some people might have added an S sticky type, or maybe they just spelled it.
Yes, make sure that it looks just like this. And remember, if you are stuck on the code, you can always get the worksheet with the code already properly typed in. I'll show you that real quick. Yeah, if you go back to the website, you can see it says worksheet with blanks filled in. If you open that one, it has the blanks already filled in. So if you like really get stuck, you can just run that. Yes, and if you're on Deposit Cloud, under answer keys, you can open that and it will, and here's the one with the blanks already filled in. You can see it has, the blanks are already all filled in there, so. Yes. So if you really get stuck, use that as a fallback option. Okay. So basically, the next step is we want to account for the fact that each sample had a different total read count. So instead of just comparing the counts between samples, we want to compare the counts basically divided by the total size of the number of reads from that sample. And usually that's given in units of counts per million. And there's a function in the packages that we're working with, I forget which one, called CPM, which will do the counts per million from the Edge R package. Yeah, most of this stuff comes from Edge R, which is a package for looking at differential expression. If we use the CPM function, it will transform the data to counts per million format based on the total reads within each sample. So I'll run that. And there's also another transformation that is often used is the log counts per million. So the counts per million on a logarithmic scale, because when you take a logarithm, then you're actually looking at, instead of looking at differences, you're looking at ratios, multiplicative difference in abundance. You know, instead of saying this sample has 10 more, reads than another, say this one has 10 times more than another. So you're looking at the log scale, which is often more appropriate for these expression data. So we're going to actually be looking at log counts per million. So if you run CPM with an additional argument, log equals true in all capital letters, log in lowercase, it will do the CPM transformation on a log scale to give you log counts per million. I haven't seen that one before. Oh, I see. It's because, yes, I think because we assign CPM to an object, but I think it should still be fine. That's just a warning. I should have given this a different name. Now there's a function and an object with the same name in the environment. Okay, Everyone good there?
Can we, I'm just trying to get them caught up.
Yes.
Quickly.
Does anyone need help? I can,
View like what is happening right after that, like,
sorry.
for example, you did like, CPM, right? And then
Yeah.
that changes your,
Yeah, I
data.
should, but that was not a good practice. I should have called this something different so that it didn't have the same name as the function. Well, here, then the next line is to run summary on this LCPM object we created. It escaped to dismiss that autocomplete thing. If we run summary on it, it will give us for each of the columns in the data, for each of the samples, it will tell us the range of the data. So you can see that now things are range from -4 to about 15 or 16 in each column. So this is basically on a log scale now. So instead of going up to thousands, it only goes up to 15 or so because it's on a log scale. Now, the next step that we're going to do, which Matt mentioned already before, is that we need to filter the data to get rid of low expression genes that are either all zero or close to zero in every sample. Like we definitely want to get rid of genes that are exactly zero in every sample. The data already contains many genes that have all zero. So those shouldn't even be in there in the first place. They're not going to have any information to tell us anything about the difference between time points because it's zero every time. But not only do we want to get rid of exactly zero, we also want to get rid of genes that have very low expression, even if it's not exactly zero in all the samples. We still want to get rid of ones that are close to zero in all the samples. How do we define close to zero? We're just going to use sort of default cutoffs here, but in the lesson text, I mentioned the cutoffs that they use and explain them. See where I'll go to that actually. Just to let's see where we're. Yes, I'll go through that in a second. But first of all, what I'm going to do is make a copy of the transcriptome data before filtering. Just so in a minute, we're going to make a figure comparing how many genes we got rid of. So this is just like for sort of illustration purposes. Oh, wait. Oh, first, yes, I skipped this line. Let's look at how many genes have zero expression in all samples. So the row sums, we want to say equals 0. We want to make a table of how many genes, how many rows in the data have 0 in all samples. Control Enter on this line. It says false 26,000, true 19,000. So actually 19,000 of the 40 and some thousand genes are all zeros for all samples. So we're just going to throw those out right off the bat because they were not expressed in any of the samples at all. So they're not giving us any information. So we definitely want to get rid of those 19,000. But out of the 26,000 that aren't all zero, there might be some that are mostly zero or mostly a very small number that we can get rid of as well, because they're not going to have biologically meaningful variation. They're so low expressed that the noise is going to be higher than any signal there is in those genes. So we want to get rid of them as well. And the way we're going to do that, okay, so first on line 69 here, we're going to make a copy of the data unfiltered so we can compare it later. And then we're going to use the function filter by, you can see that the autocomplete comes up filter by expression. So you can actually go down and hit tab to autocomplete that function name, and it'll give you the right capitalization and stuff. Filter by expression. We're going to filter the transcriptome data. And the group argument tells us what the groups in the data are. And the reason for that is that if you go to the lesson text, the basically by default, these are the cutoffs that it's using to determine which genes to filter out. Basically, it is taking genes that by default, the minimum read count to retain those in the data would be 10 reads. And then basically, if you convert, given that the average library size is 21 million in the data set, 10 reads is roughly equivalent to 0.48 reads per million. And basically, we have 6 replicates per group, and so the filtering is keeping all genes that meet that threshold in 6 or more of the samples altogether, because there's 6 replicates per group. That's the filtering that's used. We could change the default minimum read count if we want to use a more strict cutoff or not. I think it depends on your data set, what you want to use, but we're just going to stick with that default here because it's sort of a sensible default. How are we doing? Is everyone good in terms of getting stuff to run?
Yeah, no, we had the musculus package was being,
Let's see.
so I just went ahead and just reinstalled and it like stuck on the installation.
I see. It does take a minute to install. That's weird. I thought that it had installed because it didn't cause an error earlier. Well,
Yeah, it was like when I went to look at the packages in the library, I didn't see it.
so I would, if you really are having a lot of trouble, I would recommend just using the demo server for today. And then we can spend time getting stuff installed later because yeah, I think, yes, that's why I created the demo server, just so that there wouldn't be a lot of trouble with installing stuff. So if you still really can't get it to work, I would recommend just doing that. But at least once you get it installed, everything should work for tomorrow too, because there's not any new packages tomorrow that we're not using today. Yes, again, yes. So yeah, let's proceed here. Basically, we're going to run this line filter by expression. And then we're going to subset the transcriptome data using this filtering criteria that we just calculated. So we're going to replace these with keep dot expression, and autocomplete will suggest this object that exists in our environment. And we're going to hit Control Enter on that line. And now if we look at the dimensions of transcriptome, we can see that it is now 14,000 and something by 18. So we went from 45,000 down to 14,000. So like about 20,000 of the genes had all zeros, and a further 10,000 or so had very low variation, were mostly zero. So we got rid of them. Okay, and then I didn't put any blanks in the next code. This is just, all this is you don't really even have to run this, but you can just look at what I run. It is just a figure showing the distribution of expression before and after filtering, just to illustrate what the filtering did. So I'm going to run all that. And you can see, okay, in the raw data before filtering, you can see there's one really, really big peak that's at a low number of log CPM, which is basically the zeros after we've done the log transformation, because the log CPM actually adds a small number to the zero before it takes the log transformation, because you can't take the log of 0. So that's why it's around -4. So the raw data has like one really big peak in the small numbers, and then you can see that little peak there. Well, after you filter, that little peak turns into like the big peak. So we actually have a more reasonable looking distribution. Like you want it to be kind of more like a bell curve, a sort of normal looking distribution-ish. So we kind of have that now in our filtering step. We've pretty much gotten rid of that big peak of zeros, and we're left with a more normal-ish distribution. Everyone good there? So yeah, we've done filtering to just get rid of the things that are close to 0. Next, we want to calculate normalization factors for the data, which will further account for the varying library sizes. And so we're going to use the function calc norm factors to do that. And the method is called TMM, which stands for, I think, trimmed mean. What is it? Let me see. I have some explanation for what it's doing here. Basically, it is yes, basically, we're looking at trying to get the distribution of expression values to be similar across all the samples to account for some samples having more than others. And you can see in the figure that like these distributions are pretty close across all the samples. So like the normalization isn't going to have that big of an effect because these look, they're more or less have the peak in more or less the same place. There's a little difference there, but we're going to account for that with our normalization, calculating normalization factors for basically every gene in every sample. We're going to hit Control Enter there. And it says, the object contains offsets which take precedence over library sizes and norm factors. So actually the data that was provided by our collaborators already has normalization factors pre-computed. So these ones are not going to be used. They're going to use the individual gene offsets. But you can see here are the normalization factors. So like basically the raw numbers are going to be multiplied by the normalization factor within each sample, but you can see a lot of them are pretty close to 1. So like most of the things, the normalization is not going to have much effect, but a few of them, like one of them was 0.43. That was the one that had like a very low read count in that sample. So the normalization will affect that one a lot, but some are pretty close to 1, so they won't be changed by it. So basically, we're just kind of working through steps that you would do if you have a single omics data. So far we've got the data, we've looked at it, we've done pre-processing steps to get rid of genes that don't have a lot of expression, that don't have useful information, and then we've done normalization to account for the different sizes of sequencing depth in each sample. Now we can actually look at the data and see if there's patterns between our 3 time points. So before we do any formal statistical analysis of that, like I said in the introductory material, you want to use like a really simple method to just look at patterns real quick before we do anything more sophisticated. And the first thing we're going to do is do a multi-dimensional scaling plot. I mentioned PCA. Multi-dimensional scaling is a similar method to PCA. It's a very simple multivariate method to look at patterns in a high dimensional data set using a low number of dimensions. Our data set, right, is 18 samples by now we're down to 20,000 genes. We have 20,000 dimensions. You can't visualize things in 20,000 dimensions. We want to reduce the dimensions to something we can actually look at. So we are going to make a 2-dimensional plot that conveys as much information from the 20,000 dimensions in 2 dimensions. So we're going to we're going to use the plot MDS function, plot capital MDS to make a multi-dimensional scaling plot. We have to run this line one because we made a two panel plot. We're going to restore the plotting window to have only 1 panel. So I'll just run this line to replace to get back to 1 panel. And then we're going to use the LCPM filtered object that we created, which is we did the log counts per million transformation, and we filtered the low expression genes. And we're going to make that plot. And this is just some code so that the labels show which time point each sample comes from, and the colors are are different by time point. So let's take a look. Hit Control Enter somewhere in this spot. So here's our, I'll zoom into that. Here's the multidimensional scaling plot. It's reduced the 20,000 dimensions into 2. You can see that the samples are very well clustered by time point. All the 0-hour ones are right on top of each other. All the 24-hour ones are pretty close. One is a little further. Most of the 4-hour ones are close, except way up there in the right top, you can see there is one of the samples is very different than all the others. And it's like, well, that's kind of a problematic sample. Like, is it, given how well everything else clusters, this is what I mentioned, that doing these simple visualizations can alert us to possible issues in the data before we do any real complicated models. So like, if you were doing this in real life, you would probably want to go in and like look back at whether something was weird about that sample. Why is it, I wouldn't say just get rid of data that doesn't fit your story, but if something does look weird, it could be a red flag that something went wrong with the sequencing, or, it might have just like very low sequencing depth or something like that. And so there might be issues with that.
So do you expect this to be the same for all of us?
Yeah, it should be, yes.
But mine is a little different, like showing the, see this,
Yeah, let's see.
even though there's a separation, right, but still.
Yeah, it looks similar, but not quite the same.
32%.
There shouldn't be any, have other
Mine
people
is different.
got, oh, really? That's interesting.
I'm getting like,
Is there some sort of
dimension
random
32%.
sample? It's flipped. It's okay, the dimensions are flipped.
no, actually you have.
But
oh,
this is
different-
not a
different-
mirror image. It's
Oh.
actually, yeah,
Oh,
so,
you have a different scale, I think.
Yeah, we have a different scale, right?
And also, I don't know why, like,
Mine is like
There's no-
16%
is that-
of the while.
If you run like a step seed or something, is there some random?
I don't think there might be a random seed in this. But it shouldn't, honestly, it shouldn't look that different.
I mean-
Every time I've run this, I've got the same.
I got
That's
that.
interesting.
So you got the same.
Yes, I mean, like, it might be a mirror image, that doesn't matter, but yours is just different though. So something you may have done differently in the pre-processing that.
Dim.
Oh yeah, what is the dim of LCPN filter again? Let's look at that.
Mine does, mine look fine.
Mine is not.
It's fine now?
Well, his key
Here we go.
here on our farm.
Yeah, yours is smaller, so you're using, somehow you're using different
Okay.
data than me.
If you missed a line, it could look
Oh,
like.
really,
But yeah, you are something, yours is different.
That's our time.
I don't know what, I mean, we'd have to go back and look through the whole thing to see what is different, but yeah. See,
but I think we can go.
yeah, I mean, we can try to diagnose that later, but
Get all the way down.
It is not wrong, something has gone wrong.
well, yes, I don't know what's going on with that.
I'm not sure with mine either.
Quentin, what if you rerun yours?
Yeah, I could try. Should come out the same, so let's see here. So if I want to rerun from scratch, what I would do is I would just restart R. And then run everything up to this point and see if I get the same result again.
Oh, it's above, the code is above that.
So right after like 73 and 74, my dimension were reduced to 33,000, after 73 step.
Yeah, so I ran it all again and get the same result. So, yeah. I think probably in the interest of time, it's not worth trying to go in and figure out what is different about the code, but I mean, I would say probably somewhere along the line, someone did something different, but,
Yeah, okay.
yeah,
Yours is fine, yours is fine.
maybe like yeah, because I mean.
So after like
using the
73 step,
the
If you go
same
to-
package versions. I don't know.
If you go to 73, line, after that, my dimension was like to do the dim transcriptome. What
I
is
see.
there.
Maybe somehow like your filter by expression is using a different default. We could look into whether or not that might be the case, but yeah.
Well, they all say this, like you said, they, if even
It
even
shouldn't
if
do that.
they kind of will all say the same thing, and I don't think it's a set seed thing either, is it? Because if you ran it again, it's the same.
Yeah,
Yeah, so-
I don't think it has to do with the random seed. Well, yeah, we don't really have time to go
Yeah.
and debug that right now, but let's, I'll try to look into it overnight and see what could be going on there. Cool. So, yeah, basically for the rest of, this particular lesson, what we're gonna be doing is actually doing a differential expression analysis to look at, so we're still looking at just a single omics data. If there are genes that are, significantly more expressed at one time point than another, and how many of them there are and what they are. That's basically what we're going to be doing the rest of the time. So the way that we're going to do that is to, we have to sort of do a little bit of setup to get that analysis going. The first thing we want to do is create a model matrix, which is basically just for each of our samples, a matrix of zeros and ones that says like if the first sample is in time point 1, it'll have a 1 in that column and a 0 in all the others. If a sample's in time point 2, it'll have a 1 in the time point 2 column and a 0 in all the others. So we have to say model matrix by the tilde means by 0 plus groups within the parentheses. So we want, basically that's going to create a matrix, a model matrix for each group. Hit Control Enter to run that. Add the column names to it, which will get rid of the word groups in each column name. And then if we run this line, it will print the design matrix in our console. You can see it's a matrix of zeros and ones, like I said. Sample 1 is in the time point 0, sample 2 is in time point 0, and so forth. And then we want to make a contrast matrix because we want to compare each pair of groups. Like we want to compare 0 versus 4 hours, 0 versus 24, and 4 versus 24 by taking the difference between each one. And so we're going to use the make contrasts function for that. And we're going to, so each one is just like that time point subtracted from another. So this will be TP underscore 0H minus TP underscore 4H, because that was the names of those were the different levels of the time point factor here. Yep, so if I hit Control Enter and run that, then we'll look at contrast matrix. You can see that it has the first contrast has a 1 for time point 0 and a -1 for time point 4, meaning that if we find a gene that has a positive value for this contrast, that means it's more expressed in time point 0 and less expressed in time point 4. But if a gene has a negative value for this contrast, that means it's more expressed in time point 4 and less in time point 0. And the same for the other pairs. Good. So the next thing we're going to do is, yes, a question on Teams, go ahead.
Yes, a question regarding those time points or like treating independent samples. Are those 18 samples like independently measured?
They are, the same cell was not measured at different times. Each time point was different cells. So
So they are not
they-
repeated measurements?
not repeated measures, no. In this particular case, they induced the fat breakdown and in a population of cells, but then sampled some of the cells at each time point, but they're new cells every time. But yeah, if we
Okay.
had repeats of the same organism or something at different times, we would have to use different statistical methods to account for that.
Mhm.
But we don't have to
Okay.
do that here.
Okay,
Yeah,
that
that's a
sounds
great
good. Thank you.
Thank you for raising that point. Great question.
Oh.
Okay, so there is a function called boom that we're going to use to do to basically, whenever we do, the differential expression is basically a linear model. It's basically an ANOVA that's looking at the variation within each time point versus between each time point. But ANOVA has to assume that the variance is roughly equal within each time point. But if we have I'm sorry, are people still good? I don't want people to fall behind.
There's a. Oh, here it's spelled wrong. That's its late and then capital C contrast.
It just shows up.
It only shows up if you write it. But it should be.
So now this is.
Yeah. But you do not try. So it's one. Right. That's the letter.
Oh.
Isn't that fine? It worked, right? Perfect. All right, we're good.
Sweet. So the next thing we have to do, like I said, is correct for the mean variance relationship, right? Because if our genes that have a higher mean expression also have higher variance, that will give us biased results. So we need to basically reweight the expression values to correct for that and get rid of that relationship. And that's what this boom function is doing. And so we're going to create an object called V for to say the results of this analysis. Transcriptome is the object running it on, and design is the design matrix, which will ultimately be used to tell us which genes are differentially expressed across the different groups. I'm going to hit Control Enter to run it. And plot equals true. You can see there is kind of this, it's kind of hard to tell in this distorted window, but if you look, there's sort of this mean variance trend, which actually, oddly enough, there's higher variance with the lower mean, I guess because there's more noise relative to the signal. So that's not ideal because there is a bit of a trend there, right? And so we shouldn't have that trend. So we can correct for that. If just to show you what the boom is calculating, you can hit Control Enter to look at the first few rows of the weights. And what this is doing is basically for every gene in every sample, it is calculating a weight based on the inverse of the variance that can be used to correct for that relationship between mean and variance. So yeah, now basically for the rest of the time here, we're going to just be doing the differential expression analysis and looking at its results. So LM fit, linear model fit, is the function that's used to do the differential expression. Basically just fitting a linear model more or less in ANOVA to see for each gene, is there significant differences between the groups relative to within, and then compare that using various adjustments to adjust for the multiple tests. So we're going to use this V object that we created, which like waits for the variance. And then we're going to use the design. Those are basically our fixed effects that we're comparing. We don't have any other effect besides time point. Escape there, hit Control Enter. It'll take a few seconds to compute that. And then we want to pass our contrast matrix that we created so that we can look at the pairwise difference between 0 versus 4, 0 versus 24, and 4 versus 24 time points. So contour.matrix is what we call the matrix of contrast we created. So we're going to run contrasts on that. And now there are various adjustment methods that are used to control for the fact that we have thousands of genes and we don't, we could find essentially significant results by chance if we just looked at every gene as an independent linear model. So one of the methods is called empirical Bayes. It basically it kind of pulls the estimates towards zero using the overall distribution of differences to account for the multiple tests we did. I won't get into the details of how that works, but so we're going to use the Bayes function to apply that correction. Capital Bayes. And run that. And then just to show you how the variance weighting that we did accounted for the mean variance trend, we can run this plot SA, which will make that same plot again after doing the weighting. You can see that now they're like, we used to have that curving trend. Now it's a nice flat line. So our weighting method has, I mean, you can still kind of see a subtle trend in there, but overall it's done quite a bit to get rid of that trend. And we can then use the function decide tests, which will tell us how many genes are significantly up or down regulated after applying this correction within each group or between each pair of groups. And we'll use summary to look at the total number. And so basically for each comparison, 0 versus 4, 0 versus 24, and 4 versus 24, how many genes are significantly down regulated, not significantly different, or up regulated? And in this case, it's 0 minus 4. So upregulated would be more expression at 0 hours and less at 4. Downregulated would be more at 4 and less at 0, just because of the way we set it up. So you can see that most of the 15,000 genes are not significantly different between any pair of time points. There's over 14,000 that are not differentially expressed, but there's a few hundred that are significantly different after this particular correction. But what at least what Matthias decided when he was analyzing this data is that that was not a strict enough cutoff because it's way too many genes to actually look at. So he wanted to, and this maybe, it depends what's relevant to your scientific question. He decided that only genes that have a log fold change of 1 or more are meaningful to look at. And if it's, and this is on the log base 2 scale. So if the log base 2 difference is 1, that means double, right? Because on a base 2 scale, like the log of 4 is 2, the log of 2 is 1. So there's a double difference there. So if two things are different by 1 on the log 2 scale, one is twice as much as the other. So we're only looking at genes that have twice as much or half as much expression in one time point versus another. And we can use the treat function to apply that cutoff. LFC for log fold change, we'll put a 1 for the log fold change of 1. And so we're not using the empirical Bayes correction anymore. We're now using just a cutoff that we decided was biologically significant for genes to be differentially expressed by a factor of 2. And we're going to use the decide tests function to look at what genes are up versus down regulated using that cutoff. I see we're almost at 12:30, but we'll finish up in 10 minutes, I think, since the rest of this is just kind of like looking at some output. And we can look at the summary of that. So now, based on that much stricter cutoff, there are much fewer genes that are significantly up or down regulated. We only have like 20 or so for the most part between each pair of time points that are significantly different up or down. So the remaining I believe, let me just look at the code here real quick. See what we have left. Yes, I think in the interest of time, I will not do the live coding part for the rest of this, because this is just kind of like making different figures, and understanding the exact code of how that works is not that important for learning purposes. So I'm just going to switch to the answer key that already has all the blanks filled in, and I'll just make a few plots, and we can go over what the plots say, but we won't worry about the code that was used to make them. I'm happy to explain it more in, later on, if that makes sense, folks. So we'll go in the answer key, and we're going to look at, basically, one way you can look at differential expression is a Venn diagram. So like we can say between each pair of time points, how many genes were different and how many were so we can basically look at, like, for each pair of time points, we can look at how many genes were differentially expressed. Once you see the Venn diagram, it'll make more sense. So we'll run this line of code. So basically this shows how many genes were significantly different between 0 and 4 hours only, but not 0 and 24. How many genes were significantly different between 0 and 24, but not the other one. And then 14 genes were significantly different between both time points. So like maybe those genes are really the ones that are important for change over time after the fat breakdown, because they were significant between both 0, 4 and 0 and 24. So they like changed after time 0 and stayed higher or stayed lower. So maybe those are more important. And then this number is the ones that were not different between any of them. So that's like the majority of them there. And we can do the same for the we can make one for 0 versus 4 and 4 versus 24 if you wanted to. But you can also make like a 3-part Venn diagram, which might be interesting. So you can see here's how many genes. Hopefully you all get the same result if you run this, but again, since maybe people's data were different, you might get different numbers than me. So these are the genes that are different. Like no genes were different between 0, 4, and 24. There was none there, but there's quite a few that were different between different pairs of time points. Yeah, so that's what that shows. And yeah, there's a couple other figures we can make. There's an upset plot, which some people say is better than a Venn diagram for showing, especially if you have more than 3. Like a Venn diagram with 3 circles is hard enough to interpret, but by the time you have 4, it's really hard to tell what's what. So anytime you have more than 3 groups, you probably don't want to do Venn diagrams. You probably want to go with an upset plot. So this plot shows us this is like how many genes were different both between 0 and 4 and 0 and 24. This is like how many genes were just different between 0 and 24, but not any of the other ones, and so forth. So it's kind of a more concise way to show than a Venn diagram, which is kind of neat. Yeah, and then there's some more plots we can make. Volcano plots. We're going to go over these some tomorrow too, but these mean difference plots are very, very similar to a volcano plot, which I'll show you what they look like. So this shows you, it gives very similar information to the Venn diagram of like genes that are highlighted in color are significantly different between 0 hours and 4 hours. These up ones are more expressed at time 0 and less at time 4. These low ones are more at time 4 and less at time 0. And it also shows you their average expression as well. So it's like it shows the mean of their expression and the difference of their expression in log fold units. So that's what that shows. And yeah, the last, there's also, you can also make a heat map, which we'll also look at more in the coming, tomorrow. You can make a heat map, which shows you basically for every, this is looking at only the, only, I put 1 through 20, it's only looking at the genes with the 20, the top 20 genes with the biggest differences between time points. You could do more than 20 if you wanted to, but I just put 20. And what this shows is like the, let's see, the red means higher expression and the blue means lower. So these are, it turns out there are quite a few genes. These are their names from the annotation that we have. These are genes that were low expressed at time 0 and then go up a lot at time 4, and maybe are still, some of them are still high expressed at 24. And there's also a few genes that were high expressed at time 0 and then go down. And so if you knew anything about mouse fat cell physiology, you might be like, oh, I know NB, you know, it's so obvious. I know nothing about it, so I don't know if it would or not, but you know, those might be important for metabolism and stuff like that. That's a heat map. And lastly, you can just pull out individual genes and just make a plot of their expression instead of trying to overcomplicate things with heat maps and stuff. So like leptin is maybe something that's very important for metabolism, and that was one of the genes, and you look, that's in the top 20 differential expression. So let's say we just wanted to pull out leptin, we can just pull out leptin and make a plot of its expression. So we think the, let's clear the graphics. Plot. There we go. So yeah, this is the leptin expression over time. We plotted each individual sample as a dot and then the median for each time point as a diamond. So you can see how in log units, log counts per million, the leptin expression drops over time. So that's yeah, that, but we just pulled out that one column of data from the original just to look at that, because it was one of the ones that maybe you want to highlight it for its biological significance. Yeah, so that concludes lesson 1, which was basically going through the process of looking at how to work with one set of omics data, processing it, filtering it, getting rid of not useful data, and then looking at differences between our groups. And yeah, I think we were on pretty good time in terms of lunch. So that was it for today. And luckily we get to use the same room again tomorrow. So we're going to meet here again at 9 o'clock, and we're not going to have as much lecturing tomorrow. We're just going to do hands-on stuff with multiomics for the whole time. So yeah, does anyone have any questions for now?
How about a comment?
Go for it.
Okay, so when you do the differential expression, like you said, you're gonna end up with like a table that has like your log 2-fold change and then your p-value and your raw p-value, right? And then you're going to run an adjustment, right, on that p-value. Something to keep in mind is like one of the reasons why you want to choose only, you know, features that are meaningful is because if you don't shrink that down, your p-value adjustment is effectively going to kill any significance. The more features there are, the more the harsher the p-value correction is going to be.
That's right.
So that's another important reason why you want to break it down into like just more meaningful features. I wouldn't get it.
Positives versus false negatives. Like
Yeah.
if you get rid of all significance, yes, you're not going to accidentally say one's different when it's not, but you'll have the opposite problem. You'll get rid of any real differences because you overcorrected. So you have to find
Yeah,
a way to moderate, balance that out.
yeah,
Cool. So let me stop the recording and all of that. And thanks to everyone on Teams for joining. We'll do it the same tomorrow. Yeah.
Thank you so much.
Thank you all.
Yeah, thank you.